show podcast. You here for the podcast?
Too ready.
Did you come for the podcast or did you come to watch me eating dinner? Love to know.
Both every time. Like that's part of the show now, right?
Identity for is
our new intro.
Yeah, or watch me blow my nose is the other thing.
That's awesome.
Or apply lipstick as you were doing it.
This is
As you're doing
chapstick.
right
Oh.
All right. We are on the air, everybody.
We're still punch drunk from hanging out in person last week.
I know.
We're jet lag. I'm telling you, you can get jet lag and not leave a time zone. It's a thing. Once you're on a plane, jet lag starts.
Sure.
Okay, world, let's do this podcast thing. I see Identity 4 in the chat room. He's not right here anymore. He's up there in Seattle now. Hello, Identity 4. 3, 2. This is TWIS. This Week in Science, episode number 716, recorded on Wednesday, April 10th, 2019. Seeing the whole thing. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with a black hole, Philippine fossils, and screwy sperm. But first...
Disclaimer. Caution. It's that thing you use when crossing a busy intersection. That instinct to look both ways. The native navigator in your head that whispers warnings when the future seems uncertain. Ignore it for a moment. Because ahead, there are certainties that no amount of caution can prevent. Ahead, there is a journey, an adventure, and a mystery to be unveiled that no amount of hesitation can predict. Ahead, there's only discovery. Discovery that will change everything you thought you knew about the future and the past. Discovery that will challenge who we think we are and what we thought we were capable of. This sort of life-changing discovery that is never temporary and yet only possible in the present moment. And caution-free discovery is just the sort of thing we promise to provide here on This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
Good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. Oh, you guys, they're not in Portland anymore. You're important, but you're not in Portland.
Yeah, you're still lucky to still be there. How did you get that gig? That's amazing.
Patreon patrons, work on it. Move us up there.
Yeah.
That's right. Bring everyone to Portland. It was sure fun last week. We had a lot of fun in the theater. What a great night. But we're back again.
Rose. That was a blast. Yeah.
So good. But this show, we keep going. So we're back again for another episode. And I'm really thankful that we are back because this week, the news, so much mind-blowing stuff. So let's just jump right into it. I have stories about, I'm going to talk about that black hole. That's just it.
Mm-hmm.
I've also got some really interesting autism test results. And if we get to it, some Chinese monkey brains.
Yeah.
It's going to be great. Justin, what'd you bring?
I have just a small, tiny story about an entirely new human that was discovered.
Love it.
That's going to be fun. A perspective on how to be of the planet, not just in it, thanks to some recently deciphered Cherokee inscriptions. And Blair, why wanting to live longer may be completely batty.
Oh.
Blair, what is in the animal corner?
Well, I brought some screwy sperm, as you mentioned before. I brought some cricket dating advice, and I brought the cat's meow.
Looks like for those of you who are watching right now, if you're listening, you can't see this, but Blair has also brought a dog.
Yes, I am dog sitting.
Everyone say hello to Bert.
This is Bert. There's Bert. Hi, Bert. And he requires constant attention, so I relocated my laptop to the couch for this show.
Very nice.
Happy to oblige.
Podcasting while pet sitting.
Yeah.
All this and more on this episode of TWIS. Okay. Jumping forward, if you have not subscribed to TWIS yet, why haven't you? We're everywhere you can find good podcasts, and we're on YouTube and Facebook. You can find information at twis.org. Okay, let's talk about the biggest news of this or possibly last century. This is huge.
What?
Yeah, the finding by an international team of researchers who took the Earth and turned it into a telescope. But they were able to image a black hole.
What?
Yeah, what am I talking about? I am talking about, honestly, one of the biggest physics achievements of this day and age. Einstein predicted certain consequences of relativity that should hold up not just where we are in our part of the universe, but also at the edges of black holes. And with his theory of relativity, this is kind of one of the final tests to be able to look at a black hole and see how light behaves around it. However, black holes are big and they're far away, and most of our telescopes were not up to the task of imaging a black hole. We have pointed the Hubble telescope in the direction of galaxies that we think contain black holes in the past, but just gotten the movement of stars around a dark area in the sky. We have seen the movement of dust and gas, the explosion of jets of material from central areas. All these things inferring that there was a black hole in a particular place. But since the telescopes haven't had quite the resolution to be able to actually look at a black hole, what the researchers did is they networked a bunch of telescopes over the surface of the Earth. And in networking all those telescopes, they were able to use interferometry to be able to create a more powerful virtual telescope. And it increased the resolution power of all of the telescopes to be on the scale of the Earth, allowing us to look much more closely at a black hole. Now, people might be thinking about other studies that we've talked about. This one going on, we've even talked with researchers about this Event Horizon telescope that was being created, and everyone said, "Oh, we're going to be looking at the black hole in the center of the Milky Way galaxy, aren't we? We're going to look at that." And the researchers did look at that black hole, Sagittarius A, at the center of our own galaxy. However, our black hole is really dynamic, and there's a lot of stuff going on there, which has made it much more complex to tease out the view of the black hole itself at this point in time. But they wanted to present what they had found. And they also looked at a black hole called M87 in a distant galaxy about 60, I think it was 60 billion light years away. It's really far away, and it's massive. It's an extremely massive black hole that is bigger than our solar system, some 60 million suns worth of black hole. It's huge. So it was kind of, and it's not too big. And so they're able to look at it and look at the way the light traveled around the black hole. And if you have not seen it yet, I highly recommend looking at Veritasium's video describing how the light moves around black holes. He does a great job describing it. But really, black holes, and Justin, you and I have talked about this before, they're not just a hole in space-time. You think of it like a sun or a star that's causing a spherical area of space
Yes.
to be affected. Now, light, if we were to shine a light from Earth directly at that black hole, it would not bounce back. Once it hit the event horizon, the gravitational pull of the black hole would just suck that light right in there, and there would be nothing on our cameras. It'd be like, where'd the light go? There's nothing there. But
Unless we were on the other side of it.
Not necessarily. So if you shoot light around the edge of the event horizon, it will be pulled around the backside of the black hole, and then that light will return affected by the gravitational pull of that black hole. And so what they ended up seeing in this interferometry when they're pointed their telescopes, this virtual telescope at this point in space, is not the black hole itself, but rather a ring of light from all these photons traveling around the black hole and coming back out the other side so that we could see them, illuminating dust and gas and other things in the area nearby. And so in any sense, this is a momentous achievement. The researchers have said that they call what we see a shadow, and it is a shadow in a sense, in that there's a black area in the middle. But when you look at the image of the black hole, it is something of a reddish ring with a black disc in the center. And that reddish ring is what the shadow is. It's not the accretion disk. It is actually a light shadow, this shadow that is being scattered by the gravitational forces of the black hole. And by looking at that light shadow, we could determine the mass and the gravitational field and the exact event horizon for this particular black hole.
So amazing.
It is so amazing.
Wow.
People have, we've talked before, like, have we ever seen a black hole? And we've seen stuff around black holes, but we haven't seen a black hole until now. Now people will forever know what a black hole looks like. And the next step is to continue studying this, but also to continue with the algorithms and the data parsing for our view of Sagittarius A, the black hole at the center of our universe. If you've seen the movie Interstellar, if you could, there's a vision of what a black hole would look like that was advised on by physicists who work on black holes. And they said, if you're going to make the black hole, this is what it needs to look like based on Einstein's theory of general relativity. This is what light's going to do when it travels around it. And if we could increase the resolution on this view of a black hole that we have, it would look very similar to the image from the movie.
So this might be a silly question, but what I'm
No
having
silly questions.
I'm having trouble figuring out. So what a camera would see and what our eyes would see if we were in front of it ourselves, is that the same or is that different?
So this is what cameras, these radio telescopes that are taking in electromagnetic information are, they are cameras. And
Right.
This is light information, and that is what our eyes would see as well. There would be a dark area surrounded by light that could be perceived.
And it's dark in the middle because it absorbs everything.
Yes. Yeah. I mean, it may be somewhat in the there are probably going to be differences when and if we ever actually get close to one of these things.
Mhm.
But at this point in time, the idea that we see this light and there's this dark area that we don't see, that dark area says everything. That
Yeah.
dark area is all the information.
And there's a little bit more information there too, because there's a brighter side in this photograph, if you will, of this black hole that we know we're catching a bit on the edge of its rotation.
Mhm.
And so that brighter bit you see down below is sort of telegraphing the angle at which the rotation of the black hole is in relationship to us. If we were seeing it from a proper profile of that rotation, you would expect that glow to be equal all the way around.
Yes,
But as we're
absolutely.
seeing it a bit on the edge, there's a brighter side and a darker side.
Mhm.
Yeah, and I really like this picture. This is amazing.
It looks like a cat eye, kind of.
It does. I mean, some people are trying to say it's like, the Eye of Sauron.
Yeah,
I don't think
yeah.
I think we've got other images that are closer to that in space.
Mhm, mhm.
But this is it's a little this is an eye into black holes, an eye into space. It is something that will now define for everyone from this point forward what a black hole looks like.
That picture might end up in a textbook.
It will end up
No
in a
doubt.
textbook.
Yeah.
Yeah.
And for all of those humans who are within earshot, we looked at a black hole.
I know.
We're at that point in history and the planet of science and everything else. It's just, we saw one. That's
And
awesome.
my favorite part of this also is not just that we looked at a black hole, but that we turned our planet into a telescope to
That's
do it.
clever. Yeah.
Yeah, that part is out of control.
Oh, clever humans.
And
You
let's
never fail to impress.
also let's go to who did the science as well and how the press conference was done. Proper credit was given to the scientists who did the work, and one of the astronomers who created this image is a 29-year-old assistant professor woman. And I apologize because I did not have time before the show to put in my notes an image to share on this video podcast, or time to put her exact name and where she's from and what she did. But she devised 3 years ago the algorithm to link this data together to be able to create this image. Yeah. And there's also some great work out there. The Chandra X-ray Observatory also observed this black hole at a similar time that the Event Horizon telescope was looking, and so they've got some really neat factors of scale imagery that are out there. So this is going to fill our imaginations and our conversations for some time to come.
That's awesome.
It's amazing. We saw a black hole. Humanity, you rock. Let's all come together.
Really? Good job, humans.
Cosmic perspective. We can do something that's just awesome and awe-inspiring.
Yeah.
And to see these things. I want to see more now. Let's get more. Moving on from that, though, let's talk about autism. So, Justin, I believe you brought a story a couple of years ago about some researchers who attempted fecal transplants on children with autism.
Yes.
Yes. Well, the results of that study are out.
Yes.
Yes.
And they're amazing.
Yes. This study published in Scientific Reports called Long-Term Benefit of Microbiota Transfer Therapy in Autism Symptoms and Gut Microbiota. These are from Arizona State University researchers, and they effectively demonstrated that the results that they talked about 2 years ago, which were short-term results, and they said, well, we did a fecal transplant. We saw that there was a difference in the gut microbiota of autistic children versus children without autism.
And as many of our stories end when we tell about this stuff, more research is needed.
Yes.
And thankfully, in this case, more research was done.
They kept the study going. And in this, so that the children had less microbial diversity in their guts than children without autism. And there were fewer benevolent or good bacteria also than there were not so good bacteria. And so they did the fecal transplant 2 years ago and saw, hey, it changed their gut microbiota. And some of the parents said, it looks like their behaviors are changing and some of their symptoms are less. 2 years later, all of the family, and this is a long-term study, all of the families reported in, all of the families kept going. Nobody dropped out of the study, which is almost is unheard of for long-term studies. They found a 45% reduction in core autism spectrum disorder symptoms, language, social interaction, and behavior at 2 years post-treatment compared to before treatment.
Wow.
So they were getting better. Yeah. Not just that they improved right after, but over the last 2 years, the children got even better. Their symptoms were lessened even more. And the gut microbial change that they saw was fairly well maintained.
Just the fact how much your gut is connected to everything else in your body is just every day, every week, I feel like we're learning more and more where when you go in for your annual checkup, the first thing is going to be to check out your microbiome and to go from there because it is all related. And this is a huge, huge thing that if you can make things a little bit easier just by adjusting your gut bacteria.
Right. And this is the reason that they went into this idea was also that not just that microbes are more and more implicated in that, but also that autistic children very often have gut problems, that there are digestive system issues in general, leaky gut and other things. And this is where a lot of links between dietary changes and other efforts have played in to autism treatments.
So there's another link that's sort of outside of this study that I find interesting as well, which is that the other end of what is typically found in the autistic spectrum is parents or the same genes, but other aspect of it is people who can handle large volumes of information very efficiently. So there's been correlation between autistic children and parents who are doctors or involved in information technology and this sort of a thing, right? So that the fact that none of the parents dropped out of reporting on the study probably is correlated to the fact that whatever their professions are allows them to maintain a financial stability to keep participating in the study, because massive life changes don't prevent them from being able to keep being involved. So there, I don't know if you didn't follow it. So there tends to be a wealth associated with information handling.
Oh, sure.
Yeah. And that allows what might be correlated to being able to continue to participate and report to a study as a thing you do along with everything else that life throws at you. It's outside of the study, but I think that's an interesting side thing to this.
But we don't know that for sure.
No, we don't know that for sure.
You don't know. You don't know.
No, but we do know for sure that, like, for instance, Palo Alto at the height of the information age was also a hotspot for autism. We know that it seems to run in families of doctors and people involved in IT and people who have been in that industry. It has a connection to information handling upon that spectrum. That's very fascinating. So it's a study within a study that hasn't been done, and I'm correlating, but speculating. That's what we do.
And go,
We do.
But with the study itself, the exciting aspect here is that like not just there was a 45% decrease in symptoms compared to baseline. At the start of the study, 83% of participants were rated as severe autism. At the end of the study, only 17% were severe.
Wow, that is
What?
transformational.
39% were mild,
spectrum,
moderate, and
this
44% were below the cutoff for
That is
mild
amazing.
ASD.
Okay, so now what we have to also ask, there's a follow-up to
And
this.
this is a small study,
Yeah,
so
yeah.
Right.
we
But we
need
still
more.
have to
This is
follow up with
promising.
who was the fecal source? Like who is the what was the
Healthy
fecal
people.
source that we all should be fecal
Yeah.
transplanting that had that transfer?
Well, but see, it's dependent on who you are. Is the problem?
Yeah.
So,
No,
like-
but there's so many. This is the thing too. It's just like it's at the very. This is what the cutting edge of science looks like. We have a result. We don't understand it.
yeah,
We don't really know.
we know it's good. There's something good there.
We got an amazing result. We have
Yeah.
no idea why. We have a lot of discovery ahead. This is what cutting edge looks like.
You have beginning and you have end, and you like what you have. You have to figure out what's going on in the middle that's
It's
making that
missing.
happen.
Beautiful.
Yeah, so they're
So
going to be
beautiful.
looking at trying to get a larger sample size, get more individuals involved. There were particular interventions that that helped to transform this MTT, they call it, as a therapeutic for autism spectrum.
So
And in terms of where they're going next, they do know that they have given particular dosing and time frame for the treatment to the patients already, and they're
Which
going
I
to
have
keep
a question
working on
about.
dosing and duration
Right.
to see if they can get even better benefits for different people, that maybe some individuals need booster treatments where
Right.
others didn't.
So I have a question. I don't know if you have the information in front of you about dosing and duration. So this, do you have that or not? Because my curiosity is about how well the microbiome transformed. Did it self-sustain, or did they require continual dosing, if you will?
It wasn't
or reintroduction?
continual. They pre-treated individuals with vancomycin, which is an
To wipe
antibiotic
it out. Yeah.
to kill. They did
Clear
a
what was that.
bowel cleanse, a stomach acid suppressant, and then the fecal microbiota transfer daily for 7 to 8 weeks. And this
Okay.
was 10 weeks of treatment total.
Oh, and that's it. And hard stop. And 2 years later, we're still saying, wow.
Yep.
Yeah. Oh, that's amazing.
So
It's
I guess we don't know yet, right? If the microbiome is directly related to autism, or if being on the autism spectrum makes you extra susceptible to
Yep.
irritation from gut issues.
right. So we don't know causality.
Right.
And this does not indicate causality in any way. What it indicates is that it is a potential for treatment, and that is all that
Right.
it indicates.
Right.
And as we said, we still need more study, but this is very promising.
Very neat. Either way, very good to know. Very
Good
exciting.
to know. And I am going to correct myself and answer the question the woman I was trying to talk about in the previous story about the black hole. Katie Bauman from the California Institute of Technology. She's an assistant professor in the CMS department. She was a postdoctoral fellow with the Event Horizon Telescope
Okay.
and an MIT grad.
It was like, actually, we don't need this telescope. What we need is an algorithm. I got it. I got this. Nice.
We got some great science being done. This is This Week in Science. Justin, what did you bring?
Let me. Oh, yeah. So from one mind-blowing set of stories to another, the braided stream is a term that we've used a few times, mentioned a bit on the show, as an analogy for human evolution. But before I get into that, here's the dictionary.com definition just so we can, because I don't think we've done this. What is meant by braided stream?
Right.
Because it's a thing. It's the actual thing. So, braided stream, a stream consisting of multiple small, shallow channels that divide and recombine numerous times, transforming at times forming a pattern resembling the strands of a braid. Braided streams form where the sediment load is so heavy that some of the sediments are deposited as shifting islands or bars between the channels. So, follow the analogy downstream a little bit. We can sort of picture humanity throughout the ages as the waters that are going and braiding and as these little island sandbars that appear within these braids as specific species of humans that existed at certain times or places, depending on how the gene flow of the streams merged and moved on. Hopefully that provided a picture of what braided stream means in terms of evolution. And while in most contexts we speak of humans as being the humans, the current humans, us, as we like to refer to ourselves, we know that there were many more experiments in humanity than just us, and that many of these were actually living on the planet at the same time. We've got to sort of run it down a short list. Neanderthals, Denisovans, some of whom we've just learned technically might not even be Denisovans, but might need to get renamed something else. Homo floresiensis, that's the little hobbit people. Homo naledi, those are those cave-dwelling hominids. Homo erectus was even around concurrent with a lot of this. Not to mention red deer cave people who we are still trying to figure out what, where, and how they fit in. And we need to find some more about that mystery there. And of course, us, right? That's about 8 hominins, depending on how you count it, on the planet at the same time. And now something else has been added to the mix.
Ooh, a new player on the field, eh?
Human 9.
What?
That's like Planet 9.
Yeah. International team of researchers uncovered remains of a new species of human in the Philippines, which is not far from Indonesia, where Homo floresiensis was found. The new species, which I'm reading and have not heard pronounced yet, so I might be getting this wrong, Homo luzonensis, named after Luzon Island. They found a 50,000-year-old fossil, which consists of not a whole lot yet. They've got an adult finger toe bones. They've got teeth, a child's femur. So they have elements of this hominin that are distinct enough to be a new hominin. So part of the problem is they can't quite tell. Like, this is Professor Piper of the Australian National University says the findings represent a major breakthrough, which is obvious now, in our understanding of human evolution. But he also goes on to say the size of the teeth generally, though not always, reflect the overall body size of a mammal. So we think Homo luzonensis is probably relatively small. Exactly how small, we don't know yet. We would need to find some skeletal elements from which we could measure body size more precisely. So really an awesome discovery. The extremities, hands, feet are remarkably australopithecine. Like, that's a really ancient human. It's like a 2 million year ago human to be finding like elements of in the Philippines 50,000 years ago. Also intriguing is the Calo Cave, where they found this. Near there, they had previously found rhinoceros rendering, rhinoceros that had been chopped up with stone tools. That discovery was dated at about 700,000 years ago. So this isn't just we have just found the physical evidence of a recent, which also coincides pretty decently with the time when we know the current modern humans were traveling to this region. But we now have, and in time frame of that, we also have for Homo floresiensis, the Hobbit people. We have some evidence of them 700,000-ish years ago. There looks like there was stone tool use 700,000 years ago, butchering rhinoceroses in the Philippines, which is also just by itself interesting that there were rhinoceroses in the Philippines. Even if you forget the rest of the story, that by itself is significant and interesting. So new humans.
This is all so fascinating. I mean, we talk about, like you said, the braided stream all the time. Not all the time, but quite often. But it's still shocking that there could have been even more diversity. Like, oh, wait.
Yeah.
So what's going on now? There's only one.
Well, but
remains.
is it? Because I mean, part of why we're only one is we think because
We're all of them.
because we think we're all of them. Well, and we've learned since. I mean, this is the adding to the story is that we find that a lot of humans have Neanderthal, and we find that a lot of Neanderthals were also part Denisovan. So, and some humans have some Denisovan direct, and then there's some combo overlap there. So it's most humans are current human, Neanderthal, and Denisovan. And, until we actually get code from Homo naledi, Homo floresiensis, and this new one, we don't know what part we are of them because we couldn't directly see what that contribution was. So the more we discover these ancient other humans in 700,000 years, Neanderthal was a significant enough of a discovery to find that there was a 400,000 year cousin that we intermingled with again.
Yeah, and
this is twice almost the age of Neanderthal in difference and persisted and coexisted with the current modern human.
Yeah, but part of it is that it was an island. And so there's this huge overturning of thought about humans evolving on islands, and very similar to Homo floresiensis is that this Homo luzonensis is shorter in stature, very smaller, more diminutive, and that this smaller size could be that kind of, not dwarfism necessarily, but this evolution on island evolution based on limited resources or limited range.
Certainly. And Homo floresiensis was very much connected to as a potential dwarfism of Homo erectus. This is going back further and saying a 2 million year old hominin might have made it to the Philippines and encountered island dwarfism over that span of time.
Yeah. And they were larger, got stuck on a log, ended up on an island, and then shrank.
And then may have concurrently coexisted with our vision of current modern humans. And because we don't yet have, because this is a recent discovery, we just found it, we don't yet know whether or not that fed back into the current gene flow. Like there's so many amazing stories within this one discovery.
Blair, you say it probably did, but if it was an isolated island population species, then it did not necessarly.
Not to maybe the largest of groups, but you also have to
the older evolutionary group potentially would have, but not that more recent luzonensis.
So I guess that's my question: is when were modern humans first found in the Philippines?
Approximately, probably in that range of about 50,000 years ago is when we started hitting those regions. So that's why this is interesting because forget everything else about the story, but just know that there's two hominins on an island, nowhere else to go,
There you go.
nobody else to dance with,
Yeah.
and try to tell me that something didn't happen.
Right. So maybe, some people might need some updates to their 23andMe profile pretty soon.
Yeah, right. We're gonna start. No, this is true. We're going to start adding a lot to the 23andMe that people might not have suspected could have possibly been there.
There you go.
Amazing.
And also that the Guinness Book of World Records smallest human is either from the Philippines or Indonesia. It's somewhere in that region. I'm just saying.
Well, that's interesting.
It's. I'm not saying it's connected, but it's probably connected. I'm not saying it's gonna be. You're saying no, and I'm saying yeah, no, but yeah.
You know what I think it's time for right now?
What's it time for?
Blair's Animal Corner.
With Blair?
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. Blair's Animal Corner.
What you got, Blair?
Oh my goodness, this is like a return to the Blair's Animal Corner of old. I have a story about mating and I have a story about sperm.
Those are two of my favorite segments on the Animal Corner.
So this first story all about sperm brings a new meaning to the term of getting screwed. That is because this is all about bird sperm shaped like a screw. This is from PhD student at University of Oslo, PhD student Hanna Nyborg Stustad. I got a little lesson
Nicely done
before we started. Thank you very much in how to pronounce that O with the slash in the middle. She wanted to look at the peculiar spiral shape of songbird sperm. She looked at 36 bird species from house sparrows to tree swallows, looking at sperm shapes. In particular, quite a few of them had spiral or corkscrew shape sperm. And they also happened to have a very high average swimming speed. Those species also tended to have high rates of abnormal or damaged sperm. So the first part of this study was published in the journal Evolution in July 2018. That was looking at the speed of screw-shaped sperm. But what just was published this week was the adjoining article looking at the association between sperm shape and sperm damage. So looking at these two studies together, it looks like there is a trade-off between swimming speed and robustness of sperm. This trade-off could help biologists to understand why there are so many sperm shapes. There are thousands of shapes of sperm in the animal kingdom. Why? They really just have one job, and they've had it forever. So why would there be this vast, vast difference? Wouldn't you expect that over evolutionary time, there would be a sperm that came about that was so good at its job? That's just how sperm was.
Streamlining. Yeah.
Streamline it. Absolutely.
So back in the class, because that's raised. I have a question.
Justin, yes.
So I don't know a whole lot of fun animal facts, but and you correct me if I'm wrong. But a duck's
this has nothing to do with their appendage.
Right.
Okay,
with their spiral.
That's like the obvious question because
this has nothing to do with the noodly appendage. This has more to do.
The intermediary organ is not involved with this.
Yeah, the intermediary organ has.
Or is that just totally? It's totally random.
So so there's an answer to that in this story, Justin.
Okay,
the reason that the sperm is corkscrew shaped, it would appear. They figured this out by collaborating with mathematicians, which I love. Like, hey, call up the math department and go, you want to help me figure out this sperm thing? So they took physical factors such as drag, viscosity, and size, and modeled all these different types of sperm shapes. And they found that the screw-shaped sperm cells moved forward when they spin. So it's as if they are drilling into female fluid. So the fluid that the sperm has to move through to reach the egg is viscous enough and thick enough, and sperm is so tiny that they actually really struggle to move through this heavy fluid. So they are screwing through this thicker fluid through the female canal to get to the egg. So in this case, that is why that's happening. With the ducks that you were asking about before, the female organ is also shaped like the male's intermediary organ. So that is just a shape matching system. So that's totally different. This looks like it's a fluid dynamics question.
Well, the fact that it was in birds. So is it just ducks that have that morphology?
That's a great question that I don't have the answer to. They are the most famous for that, but
Yeah.
we can dive into that question in the after show perhaps. But in this case with the sperm, they are drilling through this liquid to get to the egg faster. But where this gets interesting is that the screw-shaped sperm is in fact more prone to abnormality and damage. This study was not able to figure out why. The best guess so far is that it's somehow related to oxidative damage, because sperm cells in particular are very prone to oxidative damage, which leads to abnormalities that have been observed in previous studies. But there's no known reason that a shape would affect risk of oxidative damage yet. So that's where the more research comes in. But what we do know for sure now is that in these animals that they were looking at, in these birds, the spiral shape, the screw-shaped sperm is faster but more susceptible to damage, which is why you end up with this huge kind of rainbow of types of sperm in the animal kingdom, because you are trying to, it looks like this is a real pro-con.
It's a race for the winner.
Right. So is it better to have more sperm that is more fast or more durable? Do you want
this goes back
a
to
select number of really good sperm, or do you want lots of sperm?
Yeah, and this goes back to the study from a few weeks ago about the sperm that was. Didn't you have a study on old sperm?
Yes.
Not the ram sperm that was reinvigorated, that was
Liquid
frozen
nitrogen.
for 50 years. That was a cool story. But there was another story that discovered that there was a difference between really old sperm and younger sperm in their viability.
Yes,
think that was the same story.
if the dad was older, yeah.
Oh.
The specimen creating the sperm, as he got older, the sperm was not as good quality. Yeah, so there's all these questions of cost benefit and weighing pros and cons, and what is it that you need for this particular strategy. There was another study that I will just mention in passing this week about the testy size versus ornamental elements in primates, and that there was a direct trade-off there. Do you need larger testes because there is sperm competition happening, or do you need ornamentation because there is direct competition for mate access happening, right? So in the animal kingdom, I mean, you have this set resource amount, you have this set energy amount, you have these set kind of parameters of what you have to work with, and where you allocate those resources can be very different for different species, because there are different challenges for different species.
And it all comes down to those costs and the benefits, and how does the balance play out?
Right.
Where are you going to use those resources?
Speaking of balance and resources, this leads in perfectly to my next story, which is all about the cricket dating scene. So this is from University of Exeter. I love this study because it was a study looking at 9 years of demographic and behavioral data from wild populations of the annual field cricket, Gyrus campestris. gryllus. I don't know if it's a hard G or a soft G, but the field cricket. In 9 years of this data, they were looking at these animals in the wild, and they wanted to look at the ratios of males to females in a given season, mating season, next to the rate of aging of the males. So even though crickets don't live very long, they only live a few weeks, they still experience physical decline as that time passes. So the question is, do you age faster or slower when there are more females around? Now, there's a leading theory about this across the animal kingdom.
Are they particularly like nagging, or like there's?
I was wondering if you were going to go there.
Particularly friendly to be around?
Like, are they chill chicks, or are they like, really pain? Okay. Anyway, the leading theory for why animals fall apart as we get old is that we use up our energy on reproduction instead of bodily maintenance. So it's this use it or lose it scenario. The main meaning of life here is to procreate. So knowing that, you're going to use up all of your energy as time wears thin, trying to procreate as much as you can. Okay, so that's kind of the main theory. If that theory was right, you would expect that when males have to compete more for females, they should age faster. Therefore, if there's more females, would they age faster or slower? They would age slower, right? Because they would have less competition. There would be more females to pick from. They wouldn't have to compete so savagely for a select group of females, right? So the population of crickets in this crickets in the Spanish Meadow, they studied this where years varied in the number of males and females in the ratio. And they found that battling with rivals is more of a drain on males than mating with more females. So their mating success increased when they had more females, and it looks like their competition decreased. So deterioration with age was measured by how much the males chirped, which was the direct measure of kind of how a cricket is doing, because they do that to attract females and warn off rivals. So if they're chirping, that means they're still feeling good enough to mate, which means they're less fragile. And
they feel chirpy.
Right. They're feeling chirpy. In years with equal numbers of males and females, males experienced rapid decline in chirping, but when females outnumbered males, there was no decline at all.
Man, if you want to live longer,
surround yourself by a bunch of women? No. Actually, it's cut down on competition is what that's really about. So if you want to totally extrapolate this to the human condition, which I would not suggest because it's crickets, I would say that it would be the stress of competition. So you have to find a way to reduce the stress in your life. That's what this is about, ultimately. And if you're a cricket, the number one stressor is trying to get that mate in the 3 weeks that you live. So my extrapolation here.
That's 3 weeks. That's it. That's all a cricket gets.
That's it.
That's all a cricket. It's a short,
big bang boom.
Short life.
Yeah, but as in most animal stress conversations that we have, stress is really, really powerful. And this is just another source of stress in the animal kingdom.
You know how I am looking forward to getting rid of some of my stress?
How?
Continuing on with the show. We are done with the first half. The second half is coming with many more stories. I've got Chinese monkey brains.
Ooh.
In the second half. Delicious. I know Justin's got some more stories. Blair as well. More stories coming back after these messages.
Explain things you've heard more than intuition. intuition. A lot of research shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
Thank you for listening to TWIS. We appreciate you being a part of our show. We appreciate you bringing us into your lives every week. If you would like to help support TWIS, there are several ways you can do that besides listening to the show and telling your friends about us. First, you can head over to TWIS.org. TWIS.org is where you will find all sorts of information about the show, the past episodes, and links to ways that you can help support us. First, you can click on the yellow donate button if you would like to donate directly to us using PayPal. Makes it quick and easy. If you would like to donate in another fashion, we use Patreon. Patreon is a crowdfunding site for creators, and Patreon gives you a quick link where you can click that says support us. You can choose your level and it will be charged at that level once per month in a recurring fashion. Ongoing. You can set it and forget it and know that you are helping keep TWIS going. Also, you can head over to our Zazzle store. The Zazzle store is where you will find all of our merchandise. That merchandise is stuff like hats and t-shirts and other goodies that have the TWIS logo all over them. If you want to wear TWIS to school, to work, out on a day of fun, share us with everyone that way, you can do it. We also have goods with artwork from previous Blair's Animal Corner calendars. Now, if you would like to also get involved, we have a button where you can subscribe. If you're not subscribed yet, click on that subscribe button. You'll be able to find links to YouTube, iTunes, and Google Play where you can subscribe and get us every single week. Yeah, we're weekly. Join us. Join this world of fun that is This Week in Science. And everyone, I really have to say thank you. Thank you for being a part of what we do. Thank you for your ongoing support. We could not do this without you.
Can't explain the things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis.
And we're back. You're listening to This Week in Science.
Yeah, we are back. And now it is time for that segment of the show that we call This Week in What Has Science Done for Me Lately? What has it done? What has it done? We ask you. And here is our letter for this week. Shane writes in to say, my wife often tells me to shorten my time in the shower, but I enjoy a nice shower. Her theory is that we shouldn't waste energy, but my supposition is that if I'm enjoying it, maybe it's not a waste. It's hard to know what to feel without data, though. A friend of mine told me that I should just look at my gas meter and see how much gas I was using. For the hot water, you know. I did that, but knowing how many cubic meters of natural gas I burned didn't give me any intuition about if it was a lot or not. And apologies for the metric measurements here. You don't have to apologize, Shane. This is a science program. It's certainly easier for calculations. I also live in the Netherlands where we use a civilized measurement system instead of a camel force per cubit or however you measure natural gas at residences in the USA these days. That quick search was not super helpful, but I did find out that natural gas is mostly methane and has a mass of about 0.7 to 0.9 kilograms per cubic meter. I realized that I remembered enough high school chemistry to figure out that methane, CH4, has a mass of about 16 and carbon dioxide, CO2, about 44. So I could do some simple multiplication and figure out how many kilograms of carbon dioxide I was emitting per shower. A little more math. It turns out that when I shower daily, it emits about 210 kilograms of carbon dioxide in a year. Putting that into perspective, It's about the same as burning 105 liters of gasoline, or as Justin would say, about 105,000 milliliters of gasoline, something like 28 gallons for Americans. Anyway, thanks to science, I can now shower it guilt-free without care for my carbon footprint. Well, if not exactly guilt-free, at least with an amount of guilt put into the proper perspective. And that's what science has done for me lately.
Nice. You can also just circumvent this entire issue, put a solar panel on your roof, and use solar power for your shower. So
Ooh, yes.
you can even reduce even more guilt and put a gray water system in your house so that you're using your shower water for other things.
Yeah.
Then you're really not hurting anybody by taking a long shower.
Look at all these productive responses. See, there are solutions to everything. Perspective is very important, but gaining additional perspectives is good as well. Yeah. I mean, but I think a lot of us, many people, I mean, I don't know exactly, but I think many people don't consider how much energy it takes to heat their hot water to take a shower. So if you take a 5-minute shower versus a 30-minute shower, not only how much water are you using, but how much energy are you using during the difference in those two showers. And it can add up over time, depending on how much you like those long, hot showers.
Absolutely. Yeah.
But I like the solar idea. I mean, you don't even necessarily need a solar panel. It just could be a system that it uses solar energy to thermally heat the water.
I have a solar shower for camping that uses that. Yeah. Yeah. But I think also it's a good reminder that in the scientific community, we're starting to recognize that, these tiny things like turning off the lights, it's better than nothing, but it's not enough to turn the tide on big, big problems. It's those larger things that we have to tackle that actually is going to make a difference, right? And that's exactly what social science has done for me personally in my conservation education realm, is recognizing that I'm not the bad guy for talking about climate change and also driving a car. That's just a recognition that there's a system that's failing there and we need to work on the system. It's not me as an individual that's messing up.
Yeah. So you can like your shower, you can like your car, but maybe we should get rid of the gas-based energy system. Maybe we should, for cars and for other energy. I mean, you can have an electric
system.
Yeah, you can have an electric water heater, but where does that electricity come from? Can it be solar, water, wind, a reusable, renewable resource?
Yeah, there's a really great
Blair,
graphic
let's blame
about
it on the system.
telling people to be environmentalists by swimming upstream is not going to get everybody on board. You need to change the direction of the stream so everyone can move with the water together in the right direction.
That was very deep.
Yeah.
Only if you're in the middle of the stream. Anyhow.
True.
True. If you would like to send in your own "What has science done for me lately" response, send me an email, Kirsten@thisweekinscience.com, or send me a message on our Facebook page, facebook.com/thisweekinscience. There, we are there. Justin, what story are you going to talk about now?
So this is researchers who have recently interpreted Cherokee inscriptions in Manitou Cave in Alabama. These are inscriptions that took place during the Trail of Tears. So this was a massive time of crisis for the Cherokees, who were being displaced from lands that they had occupied for millennia. Quotey voice of Jan Simic, President Emeritus of the University of Tennessee System and Distinguished Professor of Sciences at UT's Department of Anthropology. These are the first Cherokee inscriptions ever found in a cave context, and the first from a cave to be translated. Talking Stones, Cherokee Sliby in Manitou Cave, Alabama. They tell us about what the people who wrote on the walls were doing in the cave and provide direct link to how some Native Americans viewed caves as sacred places. So part of it is a description of what is referred to as a ceremonial event. what we would in modern terms put into context of sports. They had a game that they played that was very much like lacrosse, with a stick and like a puck ball kind of a thing. And it's sort of interesting. One of the transcriptions kind of speaks to the level of sport that took place in 1828. We who are those that have blood come out of their nose and mouth. So this was not, yeah, not just.
Wow.
This was a game of fierce competition. But there's another aspect of this. There's a ceiling inscription written into the stone that is written backwards. And this is Simic again. Part of the inscription which reads, "I am your grandson," written backwards into the stone. And the takeaway from this is that the Cherokee who inscribed this was talking to the earth as if it was a predecessor and writing it backwards so that it could be legible from within the earth. I found this to be just an incredibly powerful perspective on how they viewed their place being of the earth and not simply here in it and or on it.
Yeah.
Right?
Yep.
But writing it so that the earth could read it from its perspective. Anyway, an incredible find, and this was also to get to the point of a transcription. There were scholars from the Eastern Band of Cherokee Indians and the United Kituwa Band of Cherokees and the Cherokee Nation of Oklahoma who contributed to being able to give a proper transcription to these cave writings, which is not a typical Native American thing, too. It was in a time of sort of being uprooted from that ancestral land, and there is also another aspect of it is writing something in stone is also sort of seeking a bit of permanence, that this message will be immortalized while everything else is in flux, transition, and chaos. So
a couple of really lost.
Yeah, a couple of really important lessons wrapped up in this otherwise simple language translation that was discovered.
What an important find! Just bringing also snapshots of that historical time, that giving us insight into the real people that were there. Yep. That's something incredible. Well, thinking about memories, this doesn't have to do with memories from the earth. This is memories in our brains. As we all know, as we get older, memory starts to falter somewhat. Well, actually April 8th, sorry about that. April 8th, in Nature Neuroscience, researchers published a study in which they used transcranial stimulation with electrical current on older brains, and they got those brains to improve their working memory to the age of, say, 20-year-olds. So this study looked at 42 people aged 20 to 29 years old and 42 people aged 60 to 76. They compared working memory tasks in the different age groups and found that older individuals were slower and less accurate at remembering things based on that working memory recall, or they also didn't identify little differences between pictograms, little pictures. When you look at those two pictures, what things are the same? What things are different? Older people had a harder time finding those differences. They also found, looking at brain activity, that the older group had less synchronization of the frontal and temporal cortices. And we've talked about on the show previously that there's this emerging hypothesis that there needs to be electrical synchronization, that the frequency of firing of the neurons and these different areas of the brain has to be synchronized for them to work together and for them to act well together in this memory formation type of way, or memory or recall. They used what is called TACS, which is transcranial alternating current stimulation for 25 minutes on all the individuals. They found that nothing really changed in the 20-year-olds, that stimulating the brains of the 20-year-olds didn't have an effect on synchronization. It didn't have an effect on their working memory. They were still just 20-year-olds. However, the group that was 60 to 76 years old, synchronization between their frontal and temporal cortices improved. And along with that improvement, there were also improvements to the working memory tasks, and also the finding the differences in the pictures task, which is something I want to be able to do when I get older. So sign me up.
So this is like getting your wheels aligned in your car. This is just like recalibrating. But can you feel it when they do this?
Yeah, but not bad. It's like slight electrical stimulation. It's
Like
electrical
tingly?
stimulation, but it's probably a little tingly.
Huh.
But
That's interesting. 25 minutes feels like a long time.
seems like it would, but I mean, you do other things sometimes for 25 minutes. I mean, and what if you were able to do the stimulation while you sat and read a book or watched television or did some other task, and it was just like, oh, time to put my thinking cap on, and you just did that. The researchers are interested in the idea that this kind of treatment could help to reduce some of the limitations of aging, but also they're really interested in how it could improve aspects of age-related dementia. And something that we have also talked about on the show before, related to frequency of firing, is an Alzheimer's treatment that uses light at a particular at a strobe light at a particular frequency that seems to improve cognitive function in Alzheimer's patients. In this study with the TACS stimulation, that it helped for a while and the effect lasted for almost an hour, but then disappeared. And so it wasn't a lasting effect. It helped the older brains act younger for a short period of time, but then they couldn't hold on to that synchronization. That something has already happened, that the neurons are like, I don't want to talk to you anymore.
I know what you're going to say. I've heard it all before.
But if you did it for an hour, would it last longer?
Right? But what I'm wondering also is, we don't really know how that light stimulation is improving Alzheimer's patients, which also does not seem to be an effect that lasts. It's something that when they treat with the strobe light, there seems to be an improvement in Alzheimer's symptoms for a short period of time, but then that dissipates as well. So maybe what's going on is the light is entraining neuronal firing that allows synchronization, which is alleviating some of these symptoms in Alzheimer's. And this is direct electrical activation going, bam, you guys talk to each other. So maybe one is direct and one is indirect in a sense. I don't know. This is I'm wondering if this is something that could be linked. It's an interesting result.
The issue with this one is that 25 minutes for an hour of benefit, that's not a very good cost benefit, right? Sitting around for 25 minutes for an hour.
So
depending on how good that hour is, it might be worth.
Right?
Yeah, sure.
So here I'm imagining I'm, going on 80, and I know I have to hang out with a bunch of 20-year-olds.
Right.
You're like, we're having lunch or something.
Yeah.
I'm going to prep. I'm going to stimulate my brain.
But then you're like Cinderella at midnight as soon as your hour's up.
Now. Yeah. I'm going to get slow.
Yeah. You're like, what's your name again? Uh-oh, I gotta go. Oh, man.
Yeah, I mean, this doesn't get at what will keep brains from losing that synchronous activity in the first place.
And
And
then
so
is there
that is the big
diminishing
question.
returns? Like, if you do this over and over again, is it less and less effective over time? Or does
Or
it
more and
keep
more.
effectiveness?
We don't
Because
know.
otherwise, just
Don't know.
put it in a hat.
Wear it
come
all the
with all this
time.
Bonnet. A big flowery bonnet.
Yeah. Or a fedora. You're like,
It's my
what's with the
tax
fedora,
bonnet.
Justin?
I
It's like,
love
oh,
it. I love it. No, I like
nothing.
it better if every old person's wearing a flowery bonnet, male or female. It
Yeah.
just doesn't matter.
Yeah. You're
It
like,
just hides-
I just thought it looked
just
nice,
hides the
but
batteries better.
how's your husband, Gerald, and your daughter, it's Christine, and your dog? Like, you got it all, right?
And,
You remember
but
everything.
it's sadly,
I
it's
remember all
just
the
listing
names.
names.
Yeah.
And you just don't stop listing names of people who are associated with that person. It just becomes an irrelevant conversation.
Yeah.
So, but this is important if we are going to figure out a way to live longer.
Yeah, I mean,
Ooh.
if we want to live longer, we want to live longer better. We
Yeah.
don't want to start our cognitive decline at 60 to 70 to 80 years old because of this desynchronous activity and then live to be 200. That's not going to work out very well. Yeah,
Yeah,
so
and
this
yeah,
is the
but-
kind of thing that you would want to stop.
But the whole idea of living longer is completely batty to begin with.
Not so much.
Yeah, no, it is. Absolutely.
I'm
According to
gonna
University of
live
Maryland researchers
forever.
who analyzed
Oh!
the evolutionary tree of bats and found that a majority of bat species they looked at the DNA of a majority of bat species and found 4 lineages that exhibit extreme longevity. Now, this is extreme longevity in terms of bats. So there's this correlation, apparently, between mammals, their size, and their longevity. We humans are pretty large mammals. And we have a lot of other things going for us that allow us to get to these 70, 80, 90-year lifespans. Elephants, 70 years is completely achievable. Orcas can be healthy, happy 50, 60 years old. There's a lot of large mammals that can exhibit longevity. If you happen to be, say, a field mouse, your longevity is in the neighborhood of 3 years. And with a reasonable graph of size to longevity, we find correlation. Pretty solid. However, this is according to Gerald Wilkinson, a biology professor at University of Maryland, lead author of this paper that was published today. However, if we lived as long as bats adjusted for size, human lifespans would be 240 years.
Yes.
So why did these animals live so long? Why are they beating this system? They looked at the DNA, tried to figure this out. So the work described in the research paper concluded that horseshoe bats, long-eared bats, common vampire bats, and at least one other lineage of mouse-eared bat live 4 times longer than other similarly sized mammals. They also found that high latitude home range and larger males than females could also be used to give a sort of predictive correlation to lifespan. So this is quotey voice of Wilkinson. Scientists are very interested in finding closely related species in which one is long-lived and one is short-lived, because it implies that there has been some recent change to allow one species to live longer. This study provides multiple cases of closely related species with varying longevity, which gives us many opportunities to make comparisons, look for underlying mechanism that would allow species to live longer. So they have what you could essentially say is the similar code of genetics within these species. And you have these outliers of longevity. And then you can sort of look and compare between these and see, is there a segment of code? Is there a segment of genetic change between these? And that's a very easy thing, at least to narrow down to pretty darn quickly. So this is a fascinating specimen of subject to study within these bats. So researchers found that hibernating bat species located in more extreme latitudes live longer than those closer to the equator. Wilkinson said the researchers can't say for sure how latitude is actually impacting longevity. Mechanism is missing, but their study suggests it may have to do with how these bats' ability to lower their temperature.
Yeah, so
are
that's
allowed
what I
their
was going to say.
temperature to
Makes
fluctuate.
perfect sense. Is the hibernation thing?
Yeah.
When you lower your body temperature, you lower your metabolism. That
And you
that would-
also lower your ability to have stress because you're like, hey, I'm sleeping.
Yes, and we know that hibernation is an excellent opportunity to heal, and that animals actually can lengthen their telomeres during hibernation, which would make perfect sense there.
Yeah, and one of the bats, which is sort of the outlier, is the vampire bat, which they're saying here, at least, the vampire bat is very unusual for a mammal in that it can let its body temperature rise and fall dramatically throughout the day. So it's almost as if
the vampire bat's basically a reptile.
Right, but yeah, kind of, but it's also like it I don't know. I saw it as like it can go through a mini hibernation state in a sense on a daily.
Mm-hmm.
Like I'm not going to spend 3 months doing this.
Right.
Part of the day, I'm going to go into this state and I'm going to spring back and do
Like
what I got to do.
estivate at noon, hibernate at night.
Yeah. And because not all adaptations extend lifespans, some actually shorten them. Being able to have this wide range within bats should help us narrow down those elements, those mechanisms that are actually important for longevity.
Interesting. And if we figure it out, we may be hibernating to live longer lives. Let's go over 200 years and sleep all the time. I like it.
Yeah, but then like, why are you staying alive? Just to be
although, and again, though, as me and Blair
awesome.
have been going back and forth, it's torpor. Is that am I saying it right?
Torpor.
Yeah. So it's not necessarily like hibernation is a really hard thing to achieve.
Yeah.
But torpor is this sort of state somewhere in between, like just having a good night's sleep and actually hibernating.
It's not quite having a good night's sleep, but it's it's adjusting your metabolic rate for sure. Yeah.
And if there were some way for humans to take on the traits of other animals, far be it from researchers to give other animals the traits of humans.
Uh-oh.
What?
Well, here come the Chinese again with a very provocative study out of the Kunming Institute of Zoology. And the researcher who led the effort, Bing Su, a geneticist, has taken human genes for brain development and transgenically inserted them into macaque monkeys.
Ooh.
This human gene is suspected to play a role in human intelligence. Now, Dr. Su, the geneticist who led this study, is known for looking into the genetics of evolutionary traits. And so the basis of this question is, at some point, humans' brains started growing and we got smarter.
Yeah.
What happened? What genes did we get? How did we change from the ancestors to who we are now? What genes led the change? And so what better thing to do than take some human brain genes and put them in macaque monkeys?
So my prediction is this study fails to produce a more intelligent macaque. And
let me tell you what the results of the study are.
Before you do, can I throw out my why I think that, and it's because of one of the theories, at least, on how humans became more intelligent was the fact that we ran bipedally everywhere, right? It's that whole forcing more blood mechanical pump of
That's one idea.
No, it is blood soaked, and you can demolish it at the end of this if this is not how the study comes out, because I haven't seen this. And over time, that little mechanical pump pushed blood up through the brain, and also the channels, the veins and the channels through which the base of the skull blood was allowed to flow to the brain increased. So my prediction would be, regardless of the genes that you change, if you haven't gone through the hard work of evolving higher blood flow to the brain, you shouldn't see a realizable effect.
Right. So is it going to turn a monkey into a human? No. But this is why they also didn't choose to do this study in higher primates. They chose to do the study in a more distantly related animal, the monkey, which is still fairly closely related, but distantly enough that they didn't think it would make a difference. Anyhow, the research generated 11 monkeys, and 5 of them survived. They gave brain measurements and discovered that each of the monkeys have between 2 and 9 copies of this human gene in their brains, and the monkeys had increased rate of brain development as they developed, leading to larger brains than monkeys that did not have the gene inserted, and also slightly higher scores on behavioral tests of intelligence. So it did work a slight bit. However,
so did they have bigger skulls and increased blood flow? Like, I'm not quite.
Right. So it's still a small sample size. This is
not
5 out of 11, which isn't even 50%. We have monkeys performing a little better on behavioral tests, and not even and having a few copies of the gene, but not necessarily having everything they would need. And when you talk about transgenic insertions, is that gene throughout the brain, or is it in all the DNA, or is it in just little bits and pieces? The researcher says that for sure the small number of animals is a limitation, but fear not, there's a solution. More monkeys. And they're testing other brain evolution genes. He's looking at another gene called SIRGAP2C, which is a DNA variant arising 2 million years ago, just when Australopithecus was leaving the scene and making way for early humans. And this particular gene
or
has
moving
been
to
called
the Philippines.
Right, or moving to the Philippines, right. And the gene has been called, quote unquote, the humanity switch, and the quote unquote missing genetic link for its role in the emergence of human intelligence early on. However, just because it's been found in fossils and modern humans, fossil DNA and modernized humans, but we don't know what the results are going to be. However, researchers also looked at the monkeys that they had inside MRI machines, and they found that the transgenic monkeys actually didn't have their brains developed faster than other monkeys, but they didn't actually have larger brains. So there was no overcrowding of the skull.
Until you increase the skull size and the blood flow, having a big brain will not help.
Not gonna help.
Actually, it would be like a constant concussion as you grew. It would be like a terrible situation. Which is why
they did do better on a short-term memory quiz.
Yeah. So, which is why I was very I was going to say I'm utterly skeptical of the result, but the idea that the development portion of it could increase, that the pace of development could increase with this gene influx, that's much more believable to me than that there could be an overall increase in intelligence based on the gene without a complete infrastructure behind it. Yeah. So, okay. Now I'm less skeptical of their result based on that.
Yeah. So it's interesting. However, there were many collaborators from around the world. And this study was first published in National Science Review it's a Beijing journal, reported by Chinese media. And now Western scientists, including one collaborator have stepped back from it and are calling the experiment reckless and question its ethics and whether or not we should be genetically modifying primates at all. Should we? And the question now, though, is the cat out of the bag, or are researchers just upset that China has taken the lead in this area of research where they are not as limited as other countries in the steps that they take in genetic experimentation?
So I guess
I would-
with some of these kind of like gray area studies, usually I feel like there's a very clear reason or goal. And for this one, I understand they're trying to find this missing link, right, of intelligence. But in terms of what you would do with that information, that's the part that I'm missing.
Oh no!
To what end are you
that would
trying to
have
figure this out?
massive implications for assisting humans in their own like developmentally disabled humans, perhaps. You could
So this
say.
is something that you might recognize
have-
if you identify it, it may be missing in certain humans. Is that
you could create
Or mutated.
have
Or
all sorts of
mutated.
therapeutical and everything else. No, it's a fascinating study. I think, but I like your take on it. My walk back of why people are walking back from it might be more along the lines of, I was really interested in this study, so I participated in it, and then I found out I might have repercussions at home in terms of being able to get grants. So
what was I doing?
I need to walk it back just so I can stay within the safety of the legal construct of the country I come from, having nothing to do with the research. That's more what I kind of heard from the necessity of walking back a study you were initially for, got a result from, and then realized, oh, we published? I thought we were just doing research. I didn't think we were publishing. Didn't know anyone was going to hear about it. I don't want it on my Google Scholar, whatever. Yeah, it's probably more along those lines.
interesting?
Yeah, it'll be interesting to see, especially, it'll be interesting to see where the research goes and whether they keep it in the monkeys or whether they advance this kind of research to primates. And will this turn into a David Brin Uplift type scenario? If any of you have ever read those science fiction novels, I highly recommend them for addressing this kind of question of lifting other species up with genetic tools. It's time for us to get to our quick stories at the end of the show. I've got 2 metal asteroids. I mean, it may sound heavy metal now when you think of asteroids, these iron asteroids, meteors that crash down. You get these iron ingots. They're heavy and they're amazing. Well, they might also have volcanoes. Researchers have come up with an idea. Yes, the researchers at UC Santa Cruz have come up with an idea that these metal asteroids may have cooled, some of them may have cooled from the outside in, leaving a hot molten interior that would cause volcanism. And so some metal asteroids may have volcanic activity, depending on how they cooled. And we have a mission heading out to an asteroid named Psyche that should be a metal asteroid, and we can check it out. So this is going to be a potential test of that hypothesis pretty soon. And then additionally, another study found that what you eat might impact the effectiveness of medications that you take, vaccines. There is a common food additive called tert-butyl hydroquinone, TBHQ, that's fairly common. It's used in frozen meat products, cooking oils, some fish fillets, processed foods like crackers, chips, and fried snacks, but not everybody puts the ingredient on their ingredient list, so you don't exactly know what it's in. But researchers with a couple of separate experiments had unvaccinated mice eating TBHQ in their food. Those that were unvaccinated and were eating this stuff had more flu virus RNA in their guts than in their lungs than in mice that didn't eat it. The TBHQ eaters also had more inflammation and increased mucus production deeper in their lungs than usual. So we don't know how this additive for food may hamper your ability to fight the flu, but this could be something that leads to differences in how flu vaccines or even just people are able to fight it off naturally. It could just affect your immune system. They think it increases an immune system protein called NRF2, and that when that gets activated, it turns down the volume on virus-fighting immune cells in the body. So you are what you eat. Eat for health. Yeah, maybe less TBHQ. We'll see. Blair, tell me about the cat's meow.
Oh my goodness. So you're a cat owner. Do you ever ask your cat to come go, "Hey, Stella"?
I do. I all the time by name
Does she come?
when she wants to.
So a recent
that's the hard
study
part.
from Sophia University in Tokyo looked at how cats recognize, or if cats recognize, certain words that we say. This was 4 experiments with 16 to 34 animals, so not a very large sample size. Each cat heard a recording of its owner's voice or another person's voice that slowly recited a list of 4 nouns or others' cats' names, followed by their name. And what they found was that cats perked up most when they heard their own name, which means they are recognizing their name. Overall, it does seem. So even though your cat may not respond when you call their name, they might not come when called, it's because they don't care. It's not because they don't know. Because they're cats.
But this gets right at the whole idea of cats. You know, cats don't care about anything human related. They don't, people give no credence to cat intelligence.
So that's part of this too, is that even with a dog, you say they know their name. They don't necessarily know their name. They know that name, that sound to mean, come over here and pay attention to me.
Right.
Mhm.
So there is.
Hold up.
It's a
How do we
particular
know that?
sequence of sounds, right? Yes.
How do we distinguish that from the sequence of sounds and response that humans have? Because this was like a funny thing that happened. I had a dog that only knew Danish. Like everything that was spoken to this dog was in Danish. Nej, ikke spise that. Don't eat that. Comedy nu. Come here now. Vil du have kiks? Do you want a treat? Like we only talked to the dog in Danish. And I mentioned this or in passing, like, yeah, my dog only understands Danish. And the person next to me went, "Wow, your dog understands 2 languages?" And I was like, no, that's not how this works.
No.
But I don't know that that's distinct from how humans learn and know things either. Like it's a sound, has a meaning.
No, it's the same. It's the same, except that because of our deep understanding of the English language, I can tell you your name is Justin, and okay, that is the proper noun that means me. What I'm saying is dogs and cats know their name, quote unquote. They recognize the sound, but they don't understand that is their proper noun for their existence.
Well,
that is the differentiation.
So now I'm going to have to
understand
test my
language,
cat.
but not grammar. Or
does Stella come to
Cappy?
Does Cappy come to Stella?
Not grammar
I
even.
need
But
to test.
So that is no. I'm saying they still know that means you come here now, and that can still mean that only means me, but it does not mean that is my name.
Right. Identification with self.
Right.
Got it.
Yes.
Yep.
Self-identification. That is the difference here.
So wait a second.
We are at the end of the show.
I know we are. But
we are at the end of the show.
There are dogs that can understand multiple names for their toys.
Correct.
And so how would you then quantify the fact that they can't understand that their name is their name, but all the names for their individual toys and items belong to those individual toys and items? That's hard to differentiate. That is
a great question. I hope everyone wants to ponder it. Yeah, I hope everyone wants to ponder it because we have come to the end of our show. Thank you, everyone, for listening. Thank you for listening. Shout outs to Fada for help with show notes and our social media. Identity 4, thank you for recording. And thanks for coming down from Seattle last week. That's awesome. Thank you to Gord McLeod and Ben Rothig. Who else is in the chat room who help with the chat room moderation. And I would love to thank our Patreon sponsors. Thank you to Paul Disney, Richard Onimus, Edward Dyer, Stuart Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Mazzaros, Jack Hall, Matthew Litwin, Jason Roberts, Bill Kersey, Bob Calder, Patrick Cohn, Eric Knapp, Richard Hendricks, Brian Condren, Dave Neighbor, Aiden Jeff, John Bertram, Mountain Sloth, Seth O'Gradney, Steve Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Corinne Benton, Sue Doster, David Wilkinson, Ben Bignell, Richard Porter, Paris Anthony, Kevin Reardon, Christoph Zuknurek, Ashish Pants, Ulysses Adkins, Sarah Chavis, Artyom Dogchev, Clifford Ramus, Paul Sampson, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Liesl Suzuki, Jim DePoe, Greg Riley, Sean Lamb, Ben Rothig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie Cohen, Gary Swinsberg, Robert Coburn, Greg Briggs, Brendan Minish, Christopher Rappin Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron E, and EO. Thank you for all of your support on Patreon. And if you would like to be a Patreon sponsor, you can find information at TWIS.org. Just click that Patreon link. Next week, we will be back once again with more science for you at 8 p.m. Wednesday evening at our twis.org/live channel, where you can join our chat room. You can also join us on YouTube live with a chat room if you'd rather be there. Both places. It's great. But if you miss it,
don't worry.
The show will still be there. We're a podcast and we're on YouTube, so it'll be archived, and you can find those past episodes on our YouTube channel and at twis.org.
Yes, thank you for enjoying the show. TWIS, as Kiki just mentioned, is also available as a podcast. Just Google This Week in Science in your iTunes directory, and we should pop right up. Or if you have one of the mobile, non-tethered type devices, you can look for TWIS, the number 4 Droid app in the Android marketplace, which, by the way, I don't actually think works anymore. We might want to look into that mentioning. Or This Week in Science in anything Apple marketplacey.
There is one thing that definitely still works, and that's our website at www.twis.org. While you're there, you can find information on anything you've heard here today. You can read show notes. You can also make comments and start conversations with the hosts and other listeners.
And the other listeners are amazing. But if you don't want to talk to them, you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, which is the name of the show, TWIS, somewhere in the subject line. Otherwise, your email will be spammed into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
Yes. But if you've learned anything from the show, try to remember.
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This week in science. I've got one disclaimer, and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's this week in science.
This week in science.
This week in science. This week in science. This week in science. I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we said, then please just remember it's all in your head. Cause it's this week in science. This week in science. This week in science. Science. This week in science. This week in science. Science. This week in 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 science.
Somewhere along the way, my computer updated and no longer opens other pages when I click on their links. What pop-up blocked? But I want to open that pop-up. You silly pop-up. I could say always, because I guess it's my website. I trust my website. You should too. Hey everyone, it's the after show. Dun dun dun. I was texting. Blair was texting. We're all texting. We're going to text each other all over the place. I think Justin got a haircut. The next episode is a palindrome. That it is. 717. Ooh. Scientific palindromes.
Yeah, remember when Justin was going to go get his haircut right before our live show in Portland?
His idea of time is an interesting thing. Yes, we said, hey, we should really get up and go over to the theater because other show. He's, what? I need to get a haircut. What? Why didn't you get a haircut before coming to Portland? I mean, we have good haircutting places here where you can have a drink and have a haircut, but get your haircut. He got his haircut. Episode 10. What are you doing flying out? It's not a palindrome. We could have, maybe I'll name next week's show Taco Cat. I will call it Scientific Taco Cat.
Well, and then it's not a palindrome anymore.
Oh, if I put scientific?
Yeah.
I guess not.
We just have to think of a palindrome with the word science in it.
That would be an interesting one.
I could do it. I could figure out something. Or I could just Google it also.
The word science backwards does not sound pretty. No. Have a good night, Fada. Oh, I'm exhausted. Today was a good day. but I'm tired. I was late this evening, which caused the show to be late this evening. As part of the science communication organization that I help run called Science Talk. I try and get money for Science Talk. And I applied for a local humanist society. They called it an innovation grant, community innovation grant. And I applied a few weeks back and I got a phone call that said, we will need you to be at our facility for a meeting to present your idea to our organization in person on Wednesday, April 10th.
Oh my.
And I went,
That's
oh.
wrong timing.
Yeah, I was like, oh. But then I was the person who submitted the proposal, and I was like, okay, we can do this. And it actually did not turn out that badly, considering. Like, I mean, I wasn't that late tonight.
No.
Yeah,
actually,
that's good.
Yeah, you were right on time.
Right on, not that bad. Yeah, ish.
You were, as I told the chat room, you were you arrived in just in time.
I did.
Which is 5 minutes late. To be fair, though, in my own defense,
I was asking for money from somebody.
In my own
It's a good
defense,
reason.
I was born 15 minutes late. So the fact that I'm only 5 minutes behind everything that happens on the planet now is actually, I've been catching up. So it's taken a while, but I'm slowly getting into the same time frame as the rest of humanity.
Do you want to hear some fun palindromes?
Sure.
Yes.
Eva, can I stab bats in a cave?
What? Eva, can I stab bats in a cave?
Yeah. This is my favorite. Mr. Owl ate my metal worm.
Wow.
I understand that these are palindromes, but they're also nonsense.
No.
They make perfect sense. You just got to read way deep into them.
Taco cat, however, that makes sense to me. That's cat, it's a taco. It's just like
a Santa lived as a devil at NASA. Also, damn it, I'm mad.
Oh, good. Damn it, I'm mad.
Never odd or even. That's good.
That's pretty solid.
Oh,
it is solid.
Doc note, I dissent. A fast never prevents a fatness. I diet on cod.
Wow.
Which you would have to do to make it a palindrome.
Oh my god. I love palindromes.
Okay, I apologize for the early exit, but I have to say goodnight, Blair.
That's okay. I am sick. I want to go to bed.
You were looking sniffly, and I too just had mentioned that I was tired because of a long day. So, hey, Santa Fe,
sounds good to me.
We'll see about what.
See some hot air balloons. Buy some turquoise. That's all I know about Santa Fe.
All right, I've never been to Santa Fe. It's not a direct flight from Portland.
No, not from here either.
Albuquerque.
Yeah. But anyway,
in the same state. Somewhere.
It is in the same state as Albuquerque. But Santa Fe would be awesome, maybe. So, okay, let's see what happens there.
All right.
And then, is there anything else that I need? We should make newsletter, Blair.
Yes. Yeah, we need to do that.
Yeah. We need to set up some things and get that going. Because we have people on a list. We made the list, and now let's send it.
Yeah, we have to flesh out the materials for sure. But but it shouldn't be too hard. We can just
especially if
Justin, you want to write a little blog? You want to write a little soliloquy?
Just send me a text reminder that I'm supposed to do something, and I'll get it done. In the meantime, though, I'm done. I gotta go.
What about oozy rat in a sanitary zoo?
That's good.
Oh.
You're breaking my
Okay, I will refrain from pushing any more twist business on you, Justin. Let's say goodnight.
Good night, Blair. Say good night, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good
Good night,
Kiki.
Kiki.
Good night, everyone. We hope to see you here next week. And yes, Ed Twitch session this Friday for sure. I look forward to it this week. Everyone out there, happy sciencing. We'll see you again soon. Thank you.